All right.
It's a 1989 or 2000.
It doesn't say, it just says
Slides for
yeah, it's the slides for today. It's called a Nova.
My car only has like 40,000
Yeah,
miles.
Nova's like a program.
How many? Were we supposed to download it?
And it's from 2000, like.
Some old lady put it in the garage for sure.
All right. Where is everybody
What
today?
the heck?
We're statistic. I'm in a statistics class.
That's
we don't
barely more than my
shit
shit.
he's doing over.
You're still listening.
I did too. Like we learned
All right.
about variance, but we didn't learn
Focus.
about
Focus. Focus. Yes, ears out. I don't know what
Working
you're
on it.
listening to, but it isn't me. And I'm an attention. I need a lot of.
You are in.
I want to say I'm an attention whore, but I didn't want to say the word whore, so I was stopping myself.
And now you said it.
Because I shouldn't speak that way. Today we're starting the next case study, so no more evolution for a little while, no more Galapagos finches for a while. This next case study is based completely in ecology. So the course is ecology and evolution, so this is an ecology section. There was plenty of ecology in the Galapagos case study as well, but this is really focused on kind of ecosystem-based ecology. Now, at the beginning of every case study, we have taken you through and gotten you hopefully familiar with the statistical techniques that are used in the paper that you're going to be reading. So the paper that you're reading for this case study is Bean et al. 2008, if I recall correctly. Just to remind you, it's been up on the Moodle site for quite some time. Let's go to the Moodle site here just briefly. If you scroll down past where we are now, which is October 14th, you will see that Bean et al. Is due the week of November 11th. It's actually due whatever the Monday of that week is, if I recall correctly. So the thing is already posted. This is the article, so you should go and read that at your leisure. Also, if you look at today's, I have put a folder with some files in it. One full file is the PowerPoint that is up on the screen right now. There's also a R script and a data file that does the analysis that I'm going to take you through today. So if you want to practice your R scripting skills and want to look at an R script that does an analysis of variance that is different from the analysis of variance that we did during the statistics workshop, you can go and look at that as well. So, two techniques that the main technique that they use in Bean et al. Is an analysis of variance. And so I want to introduce you to the basic underlying way in which a single factor analysis of variance works. Because if you can do a single factor analysis of variance, you can do 2 factors or 3 factors or 20 factors for that matter. The one that they use in the text, in the case study, in the paper that you're going to read, is actually a 2-way analysis of variance, and I'll go through their experimental design with you at the end of class today. But once again, the basic ideas underlying a 2-way ANOVA are also true for a 1-way ANOVA. And then the other thing that you need to understand to get what they're talking about statistically in this paper is what the coefficient of variation is. And that's just a way of handling variation that is scale dependent, and I'll briefly explain to you how that works. So when you do an analysis of variance, remember, normally, if you just have a single variable with 2 levels, you can do something like a t-test. But if you have a single variable with multiple levels, you can't do a t-test. You have to do something more than a t-test, because a t-test is just designed to compare 2 samples to one another. So this is the stupidest textbook example on the planet, but the nice thing about it is that it's relatively simple and straightforward. And so what these researchers were doing is they were looking at the growth of pea plants. I'm going to erase this so you don't confuse the fact that Dean might be working with pea plants. So they just used pea plants, and they watered them with 5 different kinds of water. In one case, the control treatment is just plain old water. In some cases, they added glucose. In some cases, they added fructose. In some cases, they added a mixture of glucose and fructose. And in some cases, they added sucrose. Those of you who remember this from chemistry, why would they choose these kinds of sugars? Why would you water a plant with sugar water anyway? What do you guys use sugar for? Energy. So glucose is a storage form of energy. We break down glucose during cellular respiration to release the energy that is contained within the carbon-to-carbon bonds of glucose. And so glucose is one of the places that we get energy. Do plants typically absorb this through their roots though, or do they get their glucose some other way?
Through photosynthesis.
They get it through photosynthesis, so they don't typically absorb sugar through their roots. They're going to be absorbing other things through their roots, things like water and nitrogen and phosphorus and a bunch of nutrients that we're going to talk about later on in this case study. But normally we think of plants generating their own glucose through the process of photosynthesis. What is sucrose?
Sugar,
It's what kind of sugar?
like table sugar.
It's like table sugar. Is it a simple sugar or is it a compound sugar?
Compound.
I hear simple, I hear compound.
Compound. It's made from glucose and fructose.
Okay, so it's a combination of glucose and fructose. A glucose molecule bonded to a fructose molecule. And so what they're doing is they're using simple sugars. Glucose is a simple sugar. Fructose is a simple sugar. The two simple sugars separate, but in a 2% concentration, and then sucrose at a 2% concentration. That is actually those two simple sugars bonded together into a double sugar. And so this is the rationale. Plants need sugar to grow. If you give them sugar in their water, will they use that sugar to grow? Glucose is the main sugar, but fructose is another kind of sugar, and sucrose is table sugar, which is just a compound of glucose and fructose. And then they planted these seeds, watered them with these different solutions, and then recorded how much they growth. And it's reported in the textbook example as growth increment, whatever that is. I didn't ever go and read the paper because I wasn't really all that interested. I think it's a weird experiment to do anyway. This is just a good numerical example. One of the nice things about this numerical example is there are 10 observations in each thing, so it's a totally balanced design. And we could calculate what the means are of each of these different treatments. So you basically have one variable here, one variable with 5 levels. The variable is the type of solution that you watered the plants with, and there are 5 levels. A control, 1% glucose, 1% fructose, 1% fructose, 1% glucose together, and then 2% sucrose. So 2%, 2%, 1% of each, and then 2% of sucrose.
Isn't 1% of each basically 2% sucrose?
No, because these aren't actually bonded together. It's glucose mixed up and fructose mixed up. They haven't been bonded together. So it's the two simple sugars that make up the double sugar, but they're not chemically bonded to one another. But you can see why they might want to do this if they're testing the effect of simple sugars versus double sugars on growth. So another way though that we could look at this is not just by calculating means and things like this, but we could actually put growth increment on the x-axis, put the different treatments on the y-axis, and then just plot the actual individual data. So this is where the data for the control fall. These are the data for the 2% glucose, 2% fructose, 1% glucose, 1% fructose, and 2% sucrose. Just by looking at this, does it look like the type of solution that you're giving your plants has an effect on the growth of the plants?
Yeah.
What's the effect? Hmm? They're not shrinking, they're all growing, it's just that some are growing faster than others.
It lowers growth increment.
Add sugar and it lowers growth increment, it looks like, especially if you're adding these simple sugars versus the control, and it also looks like adding double sugar might actually lower it relative to control. What else do you notice about these data distributions?
Lower variance in the ones where we had added sugar in the control group.
Okay, so the control group looks more variable than the other groups, and Jonas is now silent for a while because he's doing all the talking. So there's way more spread in the data for the control than there is for any of these other treatment levels. Now we could go through and calculate the sample means, the sample mean within each of these different treatments. And so the mean for this one is here, and this is the 95% confidence interval. The mean for that one is there with the 95% confidence interval, mean and confidence interval, mean and confidence interval, mean and confidence interval. And these are basically plotted on there because I went and I've already calculated the means and everything for all of this. And it does look like these variances, these confidence intervals are all about the same, and this one is bigger. Now before, when we talked about decision making in statistics, how do we determine whether two sample means are different from one another? Somebody other than Jonas. Yeah,
If they're outside the range of the others, no.
we'll explain that a little better.
Like, I don't know, like when we were talking about the coots and they were outside the range, the like standard deviations of the other means.
Is it a question of overlapping confidence intervals, or is it a question of inclusion of means in other confidence intervals?
Inclusions of means in other concepts.
Okay, thank you. So you might say, well, this mean is not different from this mean because we could fit this mean into the confidence interval of this mean. So when we look at data where we have means and 95% confidence intervals plotted, we can actually ask, to what degree do these means fit within the confidence intervals of other means? And we can make this kind of qualitative decision of what's different from what. And it looks like this is different from all of these, this is different from all of these, but this is also different from that. And it looks like these aren't really all that different from one another, with the exception of 2% glucose might be a little bit bigger than 1% glucose, 1% fructose. So intuitively, this is what a 95% confidence interval does, is it tells you what are the chances of you finding other sample means that are similar to the sample mean that is from this distribution. To what degree could you have gotten this sample mean from this treatment effect or not? To what degree could you have gotten this sample mean from this treatment effect or not? And so that's intuitively what an ANOVA is going to be telling you, is are these means different from one another based on what's the position of the mean relative to how much variation there is around the mean, and we quantify that with the 95% confidence interval. Now, if you have done any statistical test before you got into this class, chances are it was a t-test. Could we use t-tests to analyze these data? How so?
Like what? You have to do multiple t-tests, so you have to do one for the sucrose, one for the glucose.
You would do a t-test comparing 2% glucose to the control. 2% fructose to the control would be another t-test. 1% glucose, 1% fructose to the control. Sucrose to the control. Glucose to fructose, glucose to glucose and fructose, glucose to sucrose, fructose to that one, fructose to that one, and that one to that one. How many t-tests is that?
One.
It is a lot. Did you follow how many it was? It's 10. You would need 10 t-tests. Now, what is the alpha level that you would use for each of those? You guys remember what alpha is?
0.5, 0.05.
Okay, we would normally set it at 0.05, 5%, essentially 5%. But what is it? How would you define it? How would you put it in plain terms?
Isn't it like the amount of air that you can add? Like, because 95% would be how much confidence you
So the 95% confidence interval is related to alpha of 0.5, but this is the type 1 error rate. What do we mean when we say that it's the type 1 error rate? What is a type 1 error? Yeah, Jackson.
It's I'm in statistics right now.
Nice.
So it's when you say, I'm trying to remember it 'cause there's type 1 and type 2. It's when you reject the null hypothesis when the hypothesis is really true, I believe.
Okay, what does this mean in language that your parents would understand? Because that actually is one way of talking about the definition of a type 1 error.
Saying that two means are different when they're not.
Okay, so in this case, we're talking about means, aren't we? It's the act of saying that two means are different when they're not. So a simpler way of thinking about it is think about what the null hypothesis is. In this case, the null hypothesis usually is that population 1 is equal to population 2. And when you say that you reject that, you're saying that there's some evidence that they're not equal. And so you're basically saying that two means are not equal when in actuality they are equal. So the null hypothesis, the reason we state it in the null hypothesis way is you can specify type 1 errors for an ANOVA where you're dealing with means, but you can also say have type 1 errors for the slopes of regression lines. Is the slope of a regression line different than 0 or not? And so you might have another hypothesis where the slope of a regression is equal to 0 versus the slope of a regression is not equal to 0. Do you have a flat line or do you have a positive slope or a negative slope? All of those represent null hypotheses and alternative hypotheses. The null we generally put with h subscript 0, and the alternative is an h with a subscript a. But this is what a type 1 error is. So what is our type 1 error rate?
0.05? 5%?
Yes, but what does it mean relative to what a type 1 error is?
The likelihood that there's a 5% chance that you're going to make a type 1 error.
So if you set alpha at 0.05, you're basically saying I'm willing to accept having a type 1 error rate of 5%. So if I did the same experiment 100 times, I just know that 5% of those I might make this error if I conclude that two things are different when they're not. So that's just kind of an acceptable error rate. Somebody mentioned, Jackson mentioned type 2 error rate.
Yes.
Okay, what is type 2 error?
When you accept the null hypothesis when the hypothesis is actually wrong.
Okay, when you accept the null when it is not correct. You basically say two means are not different from one another when in actuality they are different. So it's just the opposite of a type 1 error. So this is one of those ideas that you need to kind of get ingrained in your brain. This idea of what a type 1 error and a type 2 error are.
Are we
If-
going to be dealing with beta at all, or no, just alpha?
Well, beta is the type 2 error rate. So beta is the type 2 error rate. And you can design experiments to control for type 2 errors, just like you can design experiments to control for type 1 errors. But think about this operationally. The lower you make alpha, the lower you make your type 1 error rate, the less likely you are to make a type 1 error. The less likely you are to say two things are different when in actuality they are the same. But what happens to the type 2 error rate as you make type 1 error smaller and smaller?
Increases.
It gets bigger and bigger, doesn't it? So if you want to only be wrong 1% of the time, what that means is that you're occasionally going to see means that are actually different that you don't conclude are different Because you're being really, really stringent about what different means. Because you want to avoid making the type 1 error rate less often, you are going to occasionally, you're going to make the type 2 error more often because you are being more stringent about how different two means need to be in order to conclude that they're actually different. Another way of thinking about this is, normally we set alpha at 0.05. That's just kind of the acceptable way of doing things. But if you are a cancer researcher and you are working on a cancer called glioblastoma, do you guys know what glioblastoma is? Anybody? Glioblastoma? No. We had a faculty member one time who was diagnosed with glioblastoma. It's basically a brain cancer, and when you get diagnosed with it, you probably are only going to live about the next 6 months. No known cure for glioblastoma. It's just a horrifying way to die in a relatively short period of time. Let's say you're a cancer researcher and you think you've developed a cure for glioblastoma. The problem with your cure for glioblastoma is it's so toxic it knocks out the cancer, but it also fries your liver in the process. So at the end, you may be cured of cancer, but you now need a liver transplant. How sure, as a researcher, do you want to be that this drug actually cures cancer?
100%.
Hmm?
100%, right?
You want to be really, 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 really sure. You can never be 100%. 100% sure, because if you want to be 100% sure, what's your type 1 error rate? Yeah, there's no way to do that. So you want to be sure enough that you may not want to set your type 1 error rate at 0.05. You may not want to set it at 0.1. You may want to set it even lower. You, the researcher, have control over this. You get to set your type 1 error rate at whatever you want. Now, you may set it at 0.5, and no journal on the planet will ever publish you because basically that's a 50% type 1 error rate, and do you want to say the two things are different and get it wrong 50% of the time? Probably not. No journal wants to publish that. But for this particular hypothetical example where you have this cancer drug that also causes all sorts of other systemic side effects, you might want to be a little more sure than a type 1 error rate of 0.05. But in the process of lowering that type 1 error rate so that you know that it actually works to cure glioblastoma, what are you going to miss?
Type 2 error rate?
You're going to miss that it might work. It might actually work. Because you might commit a type 2 error saying that it doesn't work when in actuality it does. And so as you run away from one type of statistical error, you run headlong into the waiting arms of the other statistical error. These two things are related to one another. So at your stage, we're just going to set it at 0.05 because that's kind of the normal accepted thing. In ecosystem ecology papers, oftentimes you'll see a relaxing of type 1 error rate. Maybe type 1 error will be 0.1 because you can sustain a 10% type 1 error rate because you're doing ecosystem ecology, you're doing experiments on these large plots of land. They're very expensive to run because you're covering large plots of land. And as a result, you may have very few samples, which then reduces your possibility of seeing differences. So you want to see those differences even if they're not as strong as they would be if you were testing at an alpha level of 0.05. You want to see those differences. You know that you have small sample sizes, so you relax your type 1 error rate. And if you make a mistake and conclude that these two treatments happening at this ecosystem scale are different, nobody dies as a result of it. The consequences of making that mistake aren't as big. And so you should think about what your type 1, Dr. Judd is laughing. What are you
Oh, no, I see
thinking?
what you mean.
Yeah.
Yeah.
One of the nice things about doing ecological work is people seldom die as a result of you making a bad call. And so you should think carefully whenever you step into research about what you want your type 1 error rate to be, because it's not set in stone, you get to choose it. Now, once you've chosen it for good scientific reasons and sample size reasons and everything else, guess what? It's set. You said, for my experiment in my system, the appropriate type 1 error rate is 0.1. You don't then get to go and change it later on when you run your statistics and find that your p-value is 0.15. If you set alpha at 0.1, this would then be the sign that you didn't achieve statistical significance. You can't tell whether or not these two means are different or not. P-value is the observed type 1 error rate based on your data. And so what you look for to make a decision about whether or not two means are different or two regression slopes are different is my observed type 1 error rate lower than my alpha level that I set at the beginning. So once you decide on what's an appropriate alpha level, it's really bad form to go and tinker with that after the fact. I was in graduate school. I was in an experimental design class, and the teacher, the professor, a guy named Patrick Phillips, was telling us about all of this, even though we should have learned this as undergraduates. We were getting it in graduate school. Again, I actually learned it in undergraduate because I took stats classes. When he did this, there was another guy in the class, and he said, well, can't you just, like, say that you wanted your alpha rate to be one thing at the beginning, and then when you get your data, look at it and then alter your alpha level? And my friend Mike Keck turned to him and said, yeah, but then again, you could just make up your data. So you want to think about this beforehand, and then once you've thought about it intelligently, you want to abide by it. Cancer drugs, where you really want to know that the drug is having a desired effect, you want to lower your alpha level. Other questions where the answer and being occasionally wrong isn't that big of a deal, you can relax that alpha level somewhat. But thinking about it beforehand is what you should be doing. So, alpha level is your type 1 error rate. How did I get into the discussion of type 1 and type 2 error rates? Well, because we have 10 t-tests. For each t-test, what is our type 1 error rate? It's 0.5. It's 0.5 for the comparison of 2% glucose to control. It's 5% for fructose to control. It's 5% for the 1-to-1 ratio of the two to the control, et cetera, et cetera, et cetera. If you do 10 t-tests, what is your observed alpha level? What is your observed type 1 error rate?
0.05 to the 10th, if you're doing 10.
You have a 0.5 alpha rate, type 1 error rate. So if you do all these, you sum them all up, and you basically get 50% type 1 error rate. Say two things are different, you have a 50% chance of being wrong. And so what ANOVA allows us to do is it allows us to look at all of these comparisons simultaneously while also maintaining an experiment-wide alpha rate, type 1 error rate of 0.05. And this is why we use ANOVA rather than just doing a bunch of t-tests.
Is ANOVA like a computer program, or is it something on a calculator, or?
You can do it by hand. I'm going to give you a, did you pick up, were those down on the view? Oh yeah. I've got a practice problem for you due on Wednesday where you'll do it by hand. Because of course, what's the best way to learn how to do something? Do it by hand. It's faster and easier to do it on a computer, but it's important that you know what the computer is doing before you go and start relying on computers. Jackson, where are you taking stats? Are you taking the psych stats class?
Yes, with Dr. Brakeman.
Yep, if you take stats with me and applied stats, every technique we use in that class, you work by hand first, because the way you learn how to do a technique is by working it by hand. You understand the underpinnings of it, how the computer is doing the calculation before you ever get a chance to rely on a computer to do it. And that's what we're going to teach you today. So it's called an analysis of variance. But you're probably thinking to yourself, well, Dr. Kolinsky, you're saying that the analysis of variance is a way to tell whether means are different from one another. Why is it not an analysis of means? I mean, you should be asking yourself that, right? We call it an analysis of variance because there are different kinds of variance. Remember that for a single sample, we gave you the equation for sample variance. What is it? Test one material. Somebody other than Jonas. Tired of hearing from Jonas today.
Sum of variables minus the square of
The sum of the individual data points minus the mean squared divided by n minus 1, the sample size minus 1. This is sample variance. This is the variance that exists around a single mean. But in this example, we have five different means. And so this variance that we're talking about now, oh, it's on the next slide. This variance is the variance within each sample. It's the difference between this data point and the mean squared plus 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 this data point minus the mean squared, 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 all added up and divided by, in this case, 9, because you have 10 data points there. So you have, in an ANOVA, this is actually within sample variance. It's the variance that is within this treatment level. It's the variance that is within this treatment level. This data point minus the mean squared, 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 this minus that squared, this minus that, this minus that, this minus that, this minus that, this minus that. All of those individuals squared, added up, divided by 9. Do the same here, do the same here, do the same here. That's all across all of those treatments. That is within sample variance. The symbol up there for within sample variance is SS within. And we mentioned this back when we were talking about this before. What does the SS stand for?
sample size.
Hmm?
Not sample size.
Nope.
Well, in statistics, is it the sum of squares?
It's the sums of squares. That's the shorthand for it. But the reason that I want to drive this point home to you is that another way of saying it, in sentence way, is it's the sum of the squared deviations of the data from the sample mean.
Data or observation. Remember we
Oh
went
yeah,
over this.
observations.
We went over this in the video you all watched in the review for the first exam.
Yeah. The difference, the deviation of the observation from the mean squared, summed. This is the sum of squares. It's the numerator part of the variance component. So really, it's just this part is the sum of squares within. You can get the mean square by dividing that by the number of degrees of freedom for that variance, for that numerator, and it will give you the mean square, but this is what the sums of squares is. It's the sum of the squared deviations from the mean. But we have lots of different means. We have 5 different means, so this is summed across all 5 of your treatment levels in this case. So that then brings us to what is sums of squares among. Sums of squares among because we're now dealing with multiple means, is the sum of the difference between each sample mean, actually that would be J now, minus the grand mean squared. It's the same concept, just now different differences. So here, if we calculated the mean of all of these observations, if we got rid of the different treatment levels and just said we have 50 observations, what is the mean of those 50 observations? The mean would be right here, because I calculated it. So I got nothing better to do with my time. And so the sum of squares among is asking, to what degree is this mean different from the grand mean squared, 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 and this mean different from the grand mean squared. And so that's what the sums of squares among is, the difference between each of the treatment means from the grand mean, and you divide that by the number of treatments minus 1. And so in this case we have 5 treatments. We would have 4 in the denominator here, because this is the sums of squares among. This whole thing is the mean squared, because it's kind of like when you calculate a mean, the mean of a sample is the sum of all of the x values divided by sample size. In this case, our sample size for treatments is 5, because we have 5 different treatments. So the degrees of freedom is always, for variances, is 1 minus whatever your sample size is. Sum squares total is what? What do you guess sum squares total is?
The sum of squares among the data and the sum of squares of different data.
That's exactly what it is. So the sums of squares among and the sums of squares within should quantify all of the variation that is in the data set. Because there's 2 forms of variation. There is a variation that is explained by differences among the different treatments. That is what is quantified here. And then there's just random variation within each of the treatments. That's what that is. Sometimes this is also referred to as residual sums of squares. Or SS resid. But it's the same thing. It's just within sample variance. When we look at this figure, there's some variation that is due to control having a different effect than 2% glucose. But then within 2% glucose, there's just some random variation within that treatment. Within control, there's just some random variation within that. Sum of the squares within quantify that random variation within a treatment level. So when we have an analysis of variance table, they're generally laid out this way. You have the number of degrees of freedom. You have among treatment degrees of freedom, which is the number of treatments you have minus 1. You have within treatment, which is the number of observations minus the number of treatments. And then the sum of the squares among is just what we went through here. The mean squared among is the sum squares among divided by the degrees of freedom. Mean squared within is the sum squares within divided by the degrees of freedom within. This should add to that, should add to that. This plus that should add to that. You can always check an experimenter's work if they give you an ANOVA table by first and foremost seeing if the numbers all add up in the table. You would be surprised at how many published ANOVA tables don't actually add up when you go and put the math to it. It's depressingly surprising how often that occurs.
Does that mean the researcher like accidentally made a mistake or just omitted data or what?
I have no idea what that means, but my girlfriend is a postdoc, and her postdoc advisor is not a big fan of putting ANOVA tables in his papers. And I'm like, yeah, but ANOVA table is your first cut on knowing whether or not the person knows what they're doing. And so she's in the habit of putting ANOVA tables usually in the electronic supplement to her papers, and Stuart's like, why are you doing that? And she's like, well, because it's transparent, and people can look at it and see if I'm actually saying the things that I'm saying, see if they're true. Any statistical result that you see in parentheses in a sentence in the text should actually be backed up somewhere by an ANOVA table that you can look at and say, okay, these numbers all add up, because if they don't add up, then something hinky is going on. Either a transcription error got made from the ANOVA table to the text, or there's a transcription error in the putting in of the ANOVA table, or something more dire has happened. I'm a big fan of seeing ANOVA table, even if it's just in the electronics supplements.
So will we be making ANOVA tables for our research?
If you do an ANOVA, yeah, I will insist on it. And since it sounds like all of you are joining the Tuesday lab, then yeah, if you have an ANOVA table, actually you can create ANOVA tables for ANOVAs, and you can actually create an ANOVA table for regression. Turns out regression and ANOVA are the same thing, we just call them different names. So yeah, you'll be doing them. Unless you're doing something like a chi-square test or something like that. I think all of you are doing either an ANOVA or regression, so yeah, we'll do it. But it's easy, the computer spits it out at you. But, you also want to check the computer. Make sure that the computer hasn't screwed something up. You should always know what your degrees of freedom are. So for any experiment that I do, I can tell you what the degrees of freedom should be like, so that when I get my output from the computer, I can look at the degrees of freedom and immediately tell myself, is there maybe a typographic error in my data set? If so, then the degrees of freedom are going to be wonky. So,
Have you ever had an error?
oh, typographic errors, like keystroking errors in inputting data? Constantly. Like you should always enter your data twice. Why? So you can subtract column from column, and if the column sums to 0, there are no mistakes in it. So yeah, typographic errors are commonplace in entering data, especially if it's a really big data set. I had a student who was working on his senior thesis data. His data set was over 8,000 lines long. Yeah, tell me you're not going to make a typographic mistake in 8,000 lines of code.
No, you are.
Lines of data. Yeah, everybody's going to make a mistake. So how do we do this? Well, we take the mean of each of these. Mean of the sum of this is 701. There's 10 of them, so the mean is 70.1. The mean of this is 59.3, 58.2, 58, 64. The number of degrees of freedom, not the degrees of freedom, the sample size is 10, 10, 10, 10, and 10, because this is a nice clean data set. Sums of squares within is the sum of 75 minus 70.1 squared plus 67 minus 71.1 squared plus 70 minus 70.1 squared. 75 minus that squared, 65 minus that squared, 71 minus that squared, 67 minus that squared, 67 minus that squared, 76 minus that squared, 68 minus that squared. That all added up is the numerator of the variance. The mean squared is the numerator divided by 1 minus the sample size. But we're not calculating that because we want to know what the sums of squares is across all 5 treatments. So you calculate the sum of squares for this one, sum of squares for that 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 one. Add those all up, you should get 245.5. That then is the sums of squares within. You've now created this value here, which is a measure of what is the total amount of variation within treatments across all 5 treatments. So you sum this up across all 5 treatments. This is sums of squares among, so we calculated this right here is just the average of all of these data points, the sum of all of these data points divided by the number of data points that you have, and that should give you a grand mean of 61.94. So to get the sum of the squares among, you would subtract 66.59 from 61.94 and square that. 6.97, sorry, 70.1 minus 61.94 squared, 59.3 minus 61.94 squared, 58 minus 61.94 squared, 58 minus 61.94 squared, 64.1 minus 61.94 squared. Those are the sums of squares of the sample means from the grand mean. Add those all up, this is the sums of squares among. And your sums of squares among and sums of squares within should all add up to be the sums of squares total. So if you add this plus 245.5, it should give you 1,322.82. So here's the sums of squares among, sums of squares within, the total sums of squares, degrees of freedom of the treatments, degrees of freedom of the within sample variance. Remember, it's the total sample size, which is 50 minus the number of treatments. That should add to 49, which is 1 minus the total number of samples you have. Yeah.
I might be missing something. How did we get to 1,077 for our total sum of squares among? That feels.
How do we get there?
Because unless I'm misunderstanding something, I thought we were just adding up 66.59, 6.97, all of those.
As a matter of fact, you are correct. But what do we have to do? In this case, all of these sample sizes are the same.
We have to square it.
We don't have to square it.
No.
What do we have to do?
Divide by 3.
We weight them by their sample size. So if you multiply each of these times 10, sorry, I should have mentioned that. Multiply each of these times 10, it should equal up to that. Because what you have to do is weight it. On your homework assignment, it has unequal sample sizes. So for these things, you have to weight each of these by how much they're contributing to the among sample variance. And so I forget, it's like 6, 8, 10, 8, and 6 or something like that is the numbers associated with those. And so you have to multiply each of these differences by the sample size that that difference was coming from. So yeah, good point. Sums of squares plus sums of squares gives you the total sum of squares among and within. Among and within should add to total degrees of freedom. The mean square is just the sums of squares among divided by the degrees of freedom. Among should be 269. The sum of squares within divided by the degrees of freedom should give you the mean square within. Because these two things are very different, what it's telling you is that there's a lot of variation due to treatment. And there's very little variation that is due to just random variation within samples. So you calculate the F statistic, which is the mean squared among divided by the mean squared within. This is variation, average variation among your treatments. This is the average variation within treatments. And remember that when you're trying to decide whether two means are different, here we have two differences among means. If there's very little variation around these two means, it's really easy to tell that mean 1 is different from mean 2. However, if there's a lot of variation around these means, it's very difficult to tell that mean 1 is different from mean 2. So what you're looking for when you run an ANOVA is to what degree is the amount of variation that is accounted for by your different treatments large relative to the amount of variation that is within your treatments. As this gets big, as mean squared among gets big, and mean square within is small, that's telling you that the treatments that you have are having an effect. If mean squared among is small and mean squared within is big, treatment is not having much of an effect. And so you would find that this is not significantly, not a significant ANOVA, and this scenario is a significant ANOVA. And so we can do that math, and we find that we have a monstrous F statistic, and we can go online and put it into a probability calculator that will give us a p-value of 0.00000000. It's 2 times 10 to the minus 6 numbers of zeros. That's how small p is. So p, which is your observed type 1 error rate, is this. Alpha was this. Is your observed type 1 error rate smaller than your predetermined type 1 error rate? Yeah, way smaller. You can say that at least 2 of these means are different with a high degree of confidence that when you say that, it's true. And the reason that you can say this, oh, there it is right there. So this is the ANOVA table that I made up. And the reason is that this one is clearly different from everybody else. This one is clearly different from these 3, and these 3 are clearly not different from one another. So the ANOVA that you get, the ANOVA table that you get, should conform to what you already see when you go and plot your data. If your ANOVA table produced an F statistic of 3.05, which would give you like a p-value of 0.9, that's showing a non-significant ANOVA, and you should not see these big differences. So your ANOVA results should always confirm with your intuition about what your data are showing you. This is just giving you the statistical support when you go to say that things are actually different. Now, we haven't done a test to see, indeed, which of these things is different from one another, but you can do that. It's a post hoc test. It's a test that you do after the fact. And when you need to do that, we'll show you how to do that. We don't really have time to do that today, but you can do that. And in reality, what you find is that this is different from everybody else, this is different from that, and all of these, and these are not different from one another. The post hoc test also gives you what you can intuitively see from the graph of the data. And this is why we graph 95% confidence intervals, so you can look at the graph and get an intuitive feel for what's different and what's not. Questions? I'm going to give you a handout before you leave that has an ANOVA with unequal sample sizes, so you're going to have to do that weighting when you calculate the mean squares yourself. But we'll get to that in a few minutes. I'm not done yet. I've still got 11 minutes, and I got something else to tell you. I got a couple more things to tell you. So anyway, take that home, work through it by hand. If you can work through it by hand, then you should be able to understand what the computer is outputting to you when you see an ANOVA table that is generated by R, when you generate those for your data in lab.
Just curious, should we
Yeah.
study the ANOVA by hand for the test?
Oh, I'm not going to give you an ANOVA by hand on the test because you wouldn't have the time to work it. Because no, that would be unreasonable in terms of time management on a test. But I might give you an ANOVA table and ask you to interpret things about the ANOVA table.
Cool.
And if you calculate the ANOVA by hand, you're going to know more about what that ANOVA table is telling you than if you don't. Yeah, Mary.
So just to clarify, the F statistic, like that stands for the treatment, like how much effect
It's the
has.
amount of total variation due to treatment effects versus divided by the total amount of variation in the data set as a whole.
Okay.
So as that amount of variation in the data set as a whole gets really big, it swamps out any differences that you might see between sample means.
Alright, and it's just referred to as the F statistic.
Yes, it's named after Fisher, R.A. Fisher, who was a brilliant guy. R.A. Fisher was a quantitative geneticist who lived back in did a lot of his work back in the 1920s and 30s. He's the one who invented what came to be known as the Breeder's equation. So the response to selection is heritability times the strength of selection. He invented that. He needed to have a way of calculating what heritability was, and sometimes you can get parent-offspring regressions. So the concept of regression was actually invented in the century before Fisher by Francis Galton, who was Darwin's cousin. He invented regression. So that he could actually quantify how relatives are related to one another. But Fisher recognized that you don't always have parents and offspring. Sometimes you just have members of families. You have siblings. And you can actually calculate heritability by comparing siblings within families and how much variance there is, how much variation there is within families compared to how much variation there is among families. And that ratio is actually used to then calculate heritability. So R.A. Fisher invented ANOVA so that he could calculate what heritability is. None of us have much chance of being as smart as R.A. Fisher was. I don't think so. Maybe I'm underestimating some of you. Other questions? We stand on the shoulders of giants who came before us, all of us do. Your case study is focused on Konza Prairie. Konza Prairie is an 8,000 plus acre experimental area south of Manhattan, Kansas. In this system, they are experimentally altering watersheds. So this is big scale science in terms of spatial scale. So each of these watersheds represents, each of these divisions represents an entire watershed. All the water that rains on this watershed flows out of this watershed at a single spot. That's kind of the definition of a watershed. In some of these cases, these watersheds are being grazed by native grazers. The native grazers are bison, so there are bison freely roaming in this green area. And the numbers refer to how frequently these areas get burned. So they get burned every year, every 2 years, every 4 years, and every 20 years. So N1A is the A replicate of a native bison area burned every year. N20A is a replicate of a native grazer watershed that is burned every 20 years. So not only is this large scale in terms of spatial scale, it is also long term in terms of the number of years that they're monitoring these watersheds. Then there are other watersheds that are ungrazed by native grazers. So this is the Kings Creek watershed burned every year, Kings Creek watershed burned every 20 years, burned every 2 years, burned every 4 years, burned every 1 year, et cetera, et cetera. So they're looking in this experiment at two major effects: the presence or absence of native grazers, and burn return frequency.
What's the guy's name again that wrote it?
Pardon?
What was the name of the paper again?
Oh, Veen et al. V-E-E-N. So a lot of times what I try and do when I'm thinking of an experiment is I try and put it in a schematic way of thinking about it. So you can think of it, and they only looked at 1 year and 4 year burns in this paper. So one way of thinking about this is that you have watersheds that are 1 year watersheds burn frequency and every 4 years burn frequency. And then they did all of this within the native grazer area because it's hard to move bison around. So what they did was they created subplots within each of these watersheds. Some of these subplots are open to grazing, and others of these subplots have fencing around them such that the grazers can't get in there. So they create these ungrazed plots within the larger grazing landscape. So they fence off little areas where the bison can't get in there and eat the grasses and forbs and everything. And so they have, within these plots, they have sampling areas, sampling area that is ungrazed, paired sampling area that is grazed. Ungrazed paired with grazed, ungrazed paired with grazed. And so in this way they can control the two factors that they're interested in, burn frequency and grazed versus ungrazed plots. And then they can look at the way these two things act independently of one another, and then also how they might have combined or synergistic or interacting effects when you have a combination of both grazing and burning at a particular frequency. And so this is now going to be a two-way analysis of variance, where you have grazer versus non-grazer and 1 year versus 4 year burns, and then you're going to have an interaction term between those, but it's the same basic underlying statistical technique of using an analysis of variance. So this is the experimental design of the paper that you're going to read. So if you pick up that paper tomorrow, you can go back to this figure and say, oh, this is what it looks like schematically. You can go onto Google Earth. Actually, are you going to give the Google Earth concept assignment?
I'm gonna show, yeah, I was, so they have to go on and look.
It has geographic coordinates, and when you go down and zoom in, you'll actually see these plots on Google Earth.
So watch Moodle for an assignment where you go on Google Earth, navigate to the spots. That'll get posted this afternoon, if it's not already up.
So then the last thing that they do in there is they use this thing called coefficient of variation to get a measure of variation. And we use coefficient of variation to test whether populations differ in their variability. And we use a coefficient of variation because sometimes variation might be correlated with the mean. So imagine if you're interested in the length of tails of organisms, and you measure the tail length of a mouse, and you measure the tail length of an elephant. Who's going to have the longer tail length? The elephant is going to have the longer tail length. Why?
It's larger.
The elephant is just overall larger, has a larger body size. But when you look at the tail length relative to the size of the animal, who has the longer tail?
No.
The mouse has the longer tail. So the same thing applies to variation. Anything that has a large mean has more potential to have a lot of variation around it simply because the mean is very large. And so you can correct for this by basically getting an estimate of the variation that takes into account what the mean is. And so it's simply the standard deviation of the sample divided by the sample mean multiplied times 100. That gives you a coefficient of variation that is now comparable across varying scales of measurement. There's a slightly modified version which accounts for some statistical peculiarities, but this version up here is the one that I want you to learn, because this one gets most directly at the idea that you're correcting variation, i.e. standard deviation, by some measure of the mean to give you a scale-dependent estimate of how variable a sample is. And so you're going to see them quantifying variation using the coefficient of variation, because they know that variation can vary with the mean value of a particular plot. And those are the two primary things that they're going to be doing. They're going to be doing ANOVAs, and sometimes they're actually going to be doing ANOVAs on the coefficient of variation, as it turns out. Questions? All right, little data set for you to play with. Turn this in on Wednesday. It won't take you that long to do it if you have a calculator. Online, on Moodle, there is a R script for analyzing the P data. If you download the R script and download the P data into the same directory, open up the R script, it will take you through doing the ANOVA, and it should produce for you the same ANOVA table that is in the PowerPoint from today, if it's functioning correctly. Oh, did I not get in? Sorry. Questions? All right, expect for some homework, Google Earth related homework from Dr. Judd.
The Google Earth homework is on Moodle, so make sure you take a look at that. Just just give yourself some time to play around and answer the questions, and should be kind of cool to see the landscape we're going to be working with.
It's really useful seeing the landscape that we're working with. Pay attention, well, there are instructions in it.
Yeah, it's got plenty of instructions. Follow along.
Follow the instructions, that's always a good thing to do.
Dr. Clancy, you're fine with meeting with all this.
Yeah, so right now? Oh, actually, I have an appointment with the
Yeah,
research
I
student.
saw.
Okay.
I'll be once again free at 4:05. Because I have a research student and I have research meeting right after this. Yes, you did. Yeah, and as soon as you guys email, as soon as possible. Yeah. We need your doctor's note.
Wait, Dr. Jensen, does that
And
mean we
then
go like
we can
back
move
tomorrow?
to December. Alright, cool.
And then
Yeah,
for like
sooner better than later.
the pollination, and I don't know what you had, but
Yeah, I'd like to meet with you all, ideally Thursday, for 20 minutes, but if
like
you
I
rep-
haven't done too much on it yet.
So you have time.